Hello and welcome back to our 8th video installment looking at biochemistry. And in this video we're going to be focused on the second half of our look at proteins. But before we do that, let's kick it off with our little summary table that we've been building on and developing throughout each of these videos. And so, remember we started our look with carbohydrates, and we said that a monomer for the carbohydrate was a monosaccharide, and we said that the bond that was formed was a glycosidic bond. And what was forming that glycosidic bond were the two functional groups, really one functional group just used twice. It was two hydroxyl groups. And so that was the first macromolecule or biomolecule that we looked at. And then from there we went into and we looked at nucleic acids, and those nucleic acids have a monomer that are nucleotides, and the type of bond that is formed to go ahead and create more complex nucleotides are phosphodiester linkage. And remember, it's the phosphate group and the 5' carbon and 3' carbons, respectively, of the nucleotides above and below. All right. And then we jumped into looking at proteins, and we said that their nucleotide, or I'm sorry, their monomers, were amino acids, and the type of bond that was created between those amino acids are peptide bonds. And remember, the two functional groups that are involved are a carboxyl group and an amino group. And so this is kind of where we have left off with our look at proteins, just kind of getting a basic understanding. And I think in the last video, I think we even touched a little bit on the shapes and the functions and the purpose of proteins. And so now we kind of want to get into a little bit more about how do these guys function. What is it that they do, and how do they do what it is that they do? And so with that, I know this is a scary slide. There is a lot of words and a lot of writing on this slide. But the key things that you need to be concerned with when we are looking at this slide, I'm going to kind of narrow this down for you just a little bit. We'll use gray. All right. First of all, you need to know what a primary protein is. Right? And a primary protein is simply a linear organization of the amino acids. Right? And so you literally have an amino acid that's attached to an amino acid, it's attached to an amino acid, attached to an amino acid, attached to an amino acid. All right. All right. And that is the chain. That is a primary protein, right, or a primary structure. It is a linear sequence of amino acids. Right. A primary protein is a linear sequence of amino acids. And of course, these amino acids are all linked to one another using peptide bonds, which we discussed their formation in the previous video. So the other thing that you need to keep in mind here about primary proteins is functionally, these guys are not active. Right? They're not active. They're not functioning. Right? So as a primary organization, the protein really doesn't do anything. It's a storage bin of information at this point in time. The next level of organization that we find within our proteins, is a secondary structure. Right? Now, a secondary structure is also not active. Right? So this protein is also not functioning. Also not active. Right? But we do see a little bit more complexity in the organization and the arrangement of a secondary protein. And that complexity comes from the fact that this primary structure becomes either coiled or folded. In other words, it will either create what we call an alpha helix, which is spring-like. Right? That would be the coiled. Right? Or it creates a beta helix, right? Which is ribbon-like or pleated-like. And so this is what that looks like. Right? Think of it like when you take a piece of paper and you fold it into a fan. Right? That's what the beta looks like. And so this is an alpha structure, and this is a beta structure. All right. Again, still not active, right? But we've got some complexity that is now going on with the overall structure. Then we have a tertiary structure. Right? Now that tertiary structure comes into two different forms: globular proteins and fibrous proteins. And this is where we start to see some activity. This is where tertiary structures will sometimes be active. Right? So they have the ability to be active. Specifically, the fibrous proteins. And so if you've got a fibrous protein, it's active. Things like collagen. Right? Collagen is, that's its most complex structure it's going to get, is a tertiary structure of protein. It's not going to get any more complex than that. Actin is another linear protein or fibrous protein. It's not going to get any more complex than that. It is active. Right? But you have globular proteins as well. And globular proteins are usually not active. And so the general rule is that globular proteins not active, while fibrous proteins are active. So it depends on the level of organization that we've got going on that will determine whether or not that tertiary structure is active or not active. And then we can take those globular proteins and join them together to form an even larger protein structure. And it's that structure that we define as being a quaternary structure. And these guys are always active. Right? Think of hemoglobin. Right? Hemoglobin is a protein that is made up of two alpha globular proteins and two beta globular proteins that are all joined together. And each of those globular proteins has a heme group, and in the middle of that heme group is an iron molecule. And that's what the oxygen attaches to. And so that's kind of where hemoglobin gets its ability to carry oxygen from. For those of you that are a little bit more visual, like myself, right, here's a visual representation of everything that was just on that previous slide. And so you can see Right? Here's your primary structure. Right? Literally, it is a linear strand of amino acids. Right? Here's an amino acid, here's an amino acid, here's an amino acid. Right? We take that linear structure and we fold it to get our secondary structure. Here's your alpha helix, here's your beta helix. Right? We can take the alpha and the beta helix, right, and we can continue to fold that into a tertiary structure, right, which will go ahead and either form a globular protein or a fibrous protein. And then we can take two of these globular proteins that you're looking at right here and link them together, two or more. All right, and so you got 1, 2, 3. You actually do have 4 globular proteins right here, right? And that makes a quaternary structure or a quaternary protein. Right? So that's a visual way of kind of looking at and gaining an understanding about what the structure of the protein can be. Right? What about enzymes? Because enzymes is the largest classification of proteins that we have. There are thousands of enzymes within the body. So what do they do? If it is such a large group of molecules, what's their function? And that's a fair question. Right? That's a fair question. Well, enzymes are proteins that supply energy to a reaction. And the reason why they supply energy to a reaction is to lower the amount of energy that you have to create, that your metabolic processes have to put out. All right, and there's some reasons for this. Right? We'll talk about what some of those reasons are here in a few minutes. Right? But first, let's just try and get an understanding of what is what exactly is involved with these enzymes. So, some things that will prevent a reaction from running, right, from moving forward. Any kind of resistance is going to slow down a process. Now, what are those resistances? Well, very often, a resistance is going to be maybe the availability of a substrate. Right? A substrate is the chemical that is going to bind to the enzyme. If you don't have enough of that substrate, the reaction won't run. Right? Well, even if you have enough substrate, if you don't have enough energy in storage, the reaction isn't going to run. And this is really the biggest hurdle that enzymes have to cross. They have to have a certain amount of what we define as being activation energy. They have to have a certain level of activation energy in order to run the reaction. So energy needed to run a reaction. Think of activation energy as a threshold. Right? If you've got hold on one second, here's a graph. Right? And on my graph, I'm going to put a curve. And my curve shows the amount of energy needed. Well, here's 0, here's 100, let's say kilocalories, whatever it is. All right. In order for this reaction to run, you must meet the threshold of energy. You need to supply this much energy before the reaction will run. Well, this creates lag time because you've got to build up that amount of energy before this reaction will run. What an enzyme does is an enzyme comes in and it says, you know what, I'm going to do some of the heavy lifting for you. I'm going to do some of the work ahead of time. All right? And so I'm going to supply you, I'm going to arrange for this much energy to be created. Now you just need to go ahead and provide that much energy. And so it reduces the barrier, it reduces the resistance to running a reaction. You have to put out less work in order to make this run. Right? So the higher the activation energy, the slower a reaction is going to run. Enzymes come in to reduce that. All right. And this speeds up the reaction. Now, there's other ways of speeding up a reaction. Now, you can speed up a reaction. Ways to speed up a reaction. All right. You could increase the heat within the body. You could do that. Right? You could raise the internal temperature of the body, and that will cause molecules to move quicker, what we call Brownian movement, and that will cause molecules to collide more, which will cause the reactions to go quicker. Absolutely. But now you've got to dissipate that heat, which means you're going to be using a lot more energy. And you got to be careful that that heat doesn't get too high, because now you can start to actually denature your proteins. You can start taking a tertiary protein and force it to unravel because of high temperature. This is risky. Right? This is risky. You could increase reactants or the substrate. Right? Sure, you could. But now you've got to store this stuff someplace. Because if you're going to increase the amount of reactants that you have, or increase the number of substrates, then you have to have a larger stockpile of it. Where are you going to keep this stuff? That's, again, not the most practical thing to do. Right? Or you could have a catalyst. And that's where the enzyme comes into play. The enzyme is going to supply some of that energy. It's going to do some of the work. And it can do that repeatedly and efficiently without increasing the heat, without increasing the need to have reactants. It will speed up the reaction and maintain homeostasis, which is exactly what we want. Right? Which is exactly what we want. Now, how does all of this tie in together? Well, enzymes have what we call an active site, right, or a binding site. And the binding site or the active site is specific for a substrate, the thing that will bind into the enzyme. And so you can see that right here. Right? Sucrose is a sugar. Right? You should now know that that is a disaccharide. Right? And there is an enzyme called sucrase that does the work of breaking apart the disaccharide so you can get to the glucose more easily. So what will happen is sucrose will bind to the active site, all right, which causes a conformational change in the enzyme. So the enzyme actually changes shape, and it hugs the substrate. That tells the enzyme we've got to fit. Right? This is supposed to be here. And then it will go ahead and basically provide the energy to do hydrolysis to break the bond. Your body doesn't have to do anything. Right? You don't have to increase anything. The enzyme is going to handle it for you. And so again, that then lowers the activation energy that is needed. All right. So it requires less of that activation energy, and that's what you're seeing right here. Right? Here's your reactants. Right? You need to supply this much energy before the reaction will run. And so now you've got to be able to supply all of this energy. Well, the enzyme comes in and says, nope, we're going to lower your output. We're going to go ahead and we're going to create the products that you need. So that way now you only have to produce this much energy and run the reaction. This is going to progress a lot quicker, and you can do more repetitions than what you see in the first one. I like to use the analogy of you want to buy a car. You've got the car picked out. The car is there. The car is $5,000. you've got $1,000 in savings, but you don't have enough for the car, and the monthly payments are a little bit higher than what you anticipated. So you got a choice. You're $4,000 short. You can go get a second job or a third job and hope you can raise the money and save the money to go ahead and put it towards the car so you can vroom vroom down the road. Right? That's a lot of activation energy. That's a lot of work to try to save that money. So you're visiting with your grandma, and if you are the firstborn, you are probably your grandmother's favorite grandchild. I'm my grandmother's favorite grandchild because I am the firstborn. And so you're sitting at her kitchen table, and she's like, are you hungry? What do you need? How's it going? You're doing the grandmom, grandchild, type of chit-chat thing. And you're like, I've got this car. It's $5,000. I really want it. I've got $1,000 saved. I'm trying to find a second and a third job to help me to save the money to be able to pay for it. And I really like the car. It's what I've always wanted. It's got the rims. It's got the radio. It's got the tint. It's got this. It is the perfect car. So Grandmom's listening to you. And Grandmom says, you know what? Because you're my favorite, I'm going to give you that $4,000. Bam! You're off to the dealership. Vroom vroom down the road. Meep meep. All right, you got it. You're done. You provided. You had the $1,000 worth of energy. Grandmom came in. She's the enzyme. She just gave you the activation energy that you were short. You didn't have to do all that extra work. That's what an enzyme does for us. That's what an enzyme does for us. And so this slide here just kind of shows you that idea of a conformational change, what we were just looking at. All right. Again, all right, the substrates must fit inside of the active site. All right. And once the substrate has fit into the active site, has bound into the active site, all right, there is a conformational change that undergoes within the enzyme that says, yes, that substrate belongs here. Right? And when that happens, whatever the enzyme is doing, in this case here, we're actually doing dehydration synthesis, we're adding a bond, right? It does that. Right? And very often amino acids work in that way. Right? You're going to find when we get into protein synthesis that there is a enzyme, right? Peptidase. Right? Peptidase is the enzyme that is going to go ahead and navigate linking one amino acid to the next amino acid, right, at the site of protein synthesis. And so we'll look at that a little bit more in chapter 3. All right. The other thing I will mention is that it doesn't always have to be a perfect fit. All right. So if you've got an enzyme and the enzyme has an active site that looks like that, right, and you've got some substrates that are floating around, and your substrates look like this. All right, here's your substrates. All right, well, this substrate not going to be able to fit. Right, not going to work. This substrate not going to be able to work. It's not going to fit in there. But this substrate, that could actually fit in there. All right? This substrate, if it turns upside down, this substrate can also fit in there. And so this is actually the premise that we use to go ahead and create a lot of our pharmaceuticals. Right? Antibiotics will go ahead and compete with bacterial substrates to go ahead and bind to certain enzymes on the membrane of the surface membrane of the bacteria. It outcompetes it. right, and prevents anything from logging into it. It's what we call competitive inhibition. Right? You are inhibiting the enzyme from working by outcompeting whatever it's supposed to go ahead and bind in there too. And so we'll look at that a little bit more as well. So enzymes, majority of the enzymes do not work in isolation. Majority of our enzymes actually work in tandem with each other. And we call that a metabolic pathway. And so what you're seeing here is an example of a metabolic pathway. So enzyme A is going to require a substrate. And that substrate is going to be substrate square. All right. So substrate square binds to enzyme A, and enzyme A produces, I don't know why I use that cyan color. I'm sorry, guys. Enzyme A allows substrate square to bind to it, and it produces product alpha. Product alpha now becomes the substrate for enzyme B. All right, and enzyme B takes substrate alpha and produces product beta. Product beta now becomes the substrate for enzyme C, right, in which enzyme C takes substrate beta and produces product lambda. And then product lambda becomes the substrate for enzyme D, and enzyme D produces the final product. So each enzyme had a role in producing an aspect of the final product. If you block any portion of this pathway, let's say you go ahead and block enzyme B. Well, enzyme A is going to do its thing. Enzyme B is blocked, and so all of this dies off, so to speak, figuratively speaking. Nothing beyond enzyme B is going to react. All right, so if you alter the enzyme, right, if you turn the enzyme on or turn the enzyme off, you can alter the pathway of the metabolic pathway and change what is being produced. You can overproduce it or you can underproduce it, which is, by the way, how a lot of our diseases work. They either shut an enzyme off, which prevents something from being created, or they turn an enzyme on that maybe overproduces something. Or they shut an enzyme off that allows a metabolic reaction to go, and now you're overproducing something. And so we can go ahead and we can use these metabolic pathways, once again, to drive the production of an end product. These guys are all working in tandem. You're not using more concentration. You're not increasing the overall temperature within the organism or the species, and you're still getting the desired outcome without having to expend as much energy. All right, and so this here is just another view of that metabolic pathway. Right, so you can see again here, enzyme A produces substrate A, which becomes, or I should say product A, which becomes the substrate for enzyme 2, which creates product B, which becomes substrate B for enzyme 3. so on and so forth. Now this end product can actually circle back and bind to any one of these enzymes and shut the system down. This is what happens in ATP, so that we don't overproduce ATP. ATP will go back and bind to an enzyme within the metabolic pathway that'll shut it down. Because remember, you can only reserve about 30 seconds worth of ATP, so it doesn't make sense to produce 3 minutes worth of ATP. You're never going to use it. You're going to waste it. And so we refer to that ability as negative inhibition. Right? So negative inhibition or negative feedback inhibition is you've produced so much of something that it's stockpiled that you inhibit the pathway from working so you can use the reserve and you bring it back down to a normal level. Then once you drop below that normal level, you reactivate the pathway, which starts to produce the end product. The end product, when it reaches a threshold, will come back around and bind to the enzymes again and shut the pathway down. So it's a negative feedback inhibition. You're using negative feedback to inhibit or activate a specific pathway. And that is one way in which we can control waste production, so to speak, right, within the enzyme. And then the last slide that I want to kind of share with you and leave with you is other ways in which we can stop an enzyme, right, from working. So we know we have negative feedback inhibition. We know we have competitive exclusion, all right, or competitive inhibition, where something else is going to compete for the binding site, and that can shut down an enzyme. But we can also change the environmental conditions that is surrounding the enzyme, and that will shut an enzyme down as well. So as I mentioned earlier, you can increase the temperature. Increasing the temperature will force an enzyme that is in an active state, either a tertiary or a quaternary state, to revert back to a non-active state. And that non-active state, when we undergo this process of going from active to non-active, that is what we refer to as denaturing the protein, right, which is making the protein not active. Right. So high temperature denatures a protein. This is exactly how a fever works. Right. At about 100 degrees, 101 degrees Fahrenheit, your body will go ahead and actually denature bacterial proteins. So the protein sites, the enzyme sites within bacteria start to break down at about 100 to 101 degrees Fahrenheit. In humans, you have to reach about 105 to 106 degrees before those proteins will start denaturing. So there's a threshold. Right? So we can impact the function of an enzyme by changing the temperature. Same thing with pH. We can alter the activity of the pH of an enzyme by either increasing the pH, making it more alkaline, or decreasing the pH, making it more acidic. That will also take and denature a protein. Again, denaturing a protein. You've got an active form of the protein. All right? You've got an active form of the protein, either a tertiary or a quaternary structure, and you're going to revert it back to a non-active state, right, which is generally a secondary or primary structure. All right. And so that is the end of our discussion on proteins. You've got one video that is left, and that last video will have us looking at lipids. And so we will start actually the next video looking at this slide here and starting to get our heads to wrap around what a lipid is. So go back, do some reviewing on this, and as always, if you have questions, jot them down. Make sure you email me, ask me during our class time what the questions are. Make sure you get them answered. pop them in a chat, whatever you need to do. And on that note, I'll catch you on the flip side.